I'm Nicole from the Brazilian National Cancer Institute, and I will tell you about an alien in a hospital data center, or the solitary management of a bioinformatics platform. The National Cancer Institute is more than a hospital. It's a reference institution for prevention and control of cancer in Brazil through integrated actions in prevention, care, teaching and research. For the benefit of research, in 2013, a very loud and hot piece of hardware landed in the hospital data center. It was the first high-performance computing equipment of the Bioinformatics platform. It was followed by the acquisition of a small supercomputing unit in 2015. And in 2018, the Dell HPC was expanded in a substantial upgrade. The IT support team, which is very competent in developing and managing corporate environment solutions, was not trained to deal with the requirements of the scientific computing world. The aliens operate in an environment based on open source software, and because the platform users are mostly students, which are not in the Active Directory of the institution, we use a separate system for registration and authentication. In addition, the nature of the data and the workload is totally different from the institutional routine. Summing up the resources of the platform, we have already more than 1,000 processing cores connected by InfiniBand network and around 300 terabytes of storage capacity. The mission of the Bioinformatics platform at INCA is to provide support for scientific projects of the Institute and its partners. To date, the Bioinformatics platform community encompasses 18 research groups, which I will refer as supergroups, carrying out 70 projects by the hands of 50 users. They are supported by 4 Godfellows from the IT team, which provide us with a huge help with technological issues. And in charge of this, one manager, technologist, system administrator, developer, scientist, head cook and bottle washer. In this context, I would like to tell you about some challenges I face and the strategies to handle them. First, considering the complexity of a multi-user environment, how can we maintain a diversity of bioinformatic tools and libraries with constantly changing requirements? One solution is to isolate environments. For the most commonly required tools and databases, we use environment models as well as shared resources to avoid redundancy. For tools demanding more complex library dependencies, we implement virtual environments and containers, and encourage users to use it according to their needs, giving them more autonomy. Another managing challenge is to set the adequate privileges for data access for authorized users, and to provide a fair share of the available resources. How do we accomplish this? With a project-centered management model, in which the principal investigator, which is called supergroup in the model, and the users are connected by a project. To illustrate this, Dr. Ada has 3 students that are working on different projects. She requests the creation of 2 projects and assigns the students according to their enrollment in each one. Dr. Charles creates one project for his students and asks Ada to help in a machine learning problem. He assigns one of her students to his project. One of his students was then invited to participate in this big sequencing project from Ada, and we will have the permission to access the project folder to perform the analysis. This intricate network is solved with traditional Linux features, based on the hierarchical folder structure and modes of file permissions. In which the supergroup folder houses the project folders. Here we are able to set storage quotas by supergroup instead of quotas by projects. The home folder will receive symbolic links to all projects to which the user was assigned. The users are instructed to avoid storing data in their home folder. To enforce this, we set a quota of 500 megabases in the home folder. The IT team helped me to implement a semi-automatic registration system, where the researcher requests the creation of supergroups, projects, user accounts, and has to agree with the platform usage and confidentiality terms. What the requester does not know is that after the form is received, I do perform a series of manual interventions until sending back a preformatted confirmation email with a temporary random password and instructions generated by an in-house pipeline prepared to give the impression of a fully automated process. With the new account created, the user can access the HPC via command line and submit jobs in the old way. But bioinformatics has evolved, and the requirements for integrative analysis of single-cell data cannot be accomplished with traditional script submissions. Users require interactive visual environments to perform dynamic analysis of complex data. At this time, we had already provided web access to an RStudio server and a local Galaxy instance. But some workflows were Python-based, and the users were asking for a Jupyter notebook that could be accessed remotely during the quarantine. To be able to answer this request, the dirty solution I found was to provide them with a script that starts a Jupyter notebook instance in a compute node, creates a tunnel to a secondary server, which will be accessed through another tunnel, allowing the user to open the notebook in the web browser. With this workaround patchwork, I finish my talk looking into the future, because the platform needs to move on and implement new management tools for providing web-based access. We also need to require users to take responsibility for the book data, create a knowledge base, and use a ticketing system, and also learn how to ask for help. With this, I would like to thank you for the attention. Thank you, the organizer, for the invitation, and thank my colleagues and my institution. Thank you very much.